Hello students, how are you? I hope you all are good. Today we are going to study a very interesting topic. So let's start it. Welcome to Online Edu Classes 2021. This is chapter number 5, the fundamental unit of life. Here, the topic which we are going to deal, cell membrane and membrane transport. So, I am your teacher, come facilitator, come educator, come presenter obviously. So, welcome to my class. Let's start it. Tell me students, how do substances move in and out of the cell? Have you thought of that? We have plasma membrane for that. It regulates the movement in and out of the cell. The membrane mainly consists of proteins and phospholipid molecules which are scattered throughout. So this is a function of plasma membrane. C. Separate cell from environment, transport substances in and out of the cell, receive and respond to stimuli, that is external environment. Coming to the properties, we have hydrophobic and hydrophilic properties both in the plasma membrane. Definitely it is selectively permeable. Coming to the structure, it is fluid mosaic model, lipid bilayer, phospholipids is 75%, cholesterol 20%, glycolipids 5%. I'll tell you one of the important facts. You all are very fond of sandwiches, I guess. So if you have a layer of sandwich, that is two breads are there, and in between you are going to put potato. So that becomes potato sandwich. So the two layers of breads are representing the bilayer and potato is in between. That is a case in case of plasma membrane's structure as well. See, this is what about the structure of plasma membrane. You can see the lipid bilayer molecules and an embedded protein molecules in between which are shown by blue color. So phospholipid, it is having two types. First is the polar hydrophilic water-loving heads, and it is also having non-polar water-fearing tails. Heads are facing out and tails are facing in. You can see inner part of the cell is represented as cytoplasm and outer part is extracellular fluid. This is one more diagram to show or to have a clear view regarding plasma membrane. Fluid mosaic model, definitely it is given by Singer and Nicholson. I hope you people know it already. This is the 3D structure. Coming to the functions of membrane proteins. We have different types of proteins in fact in plasma membrane. They are peripheral or integral. Peripheral protein means which are outside and integral protein means which are inside. We have ion channels, we have carriers, we have receptors, linkers, cell identity markers. All these things are present in a plasma membrane. See, these are the peripheral proteins and the second diagram represents the integral protein. Coming to the membrane permeability. Selectively permeable membrane, definitely it is. Only some substances can pass through this. That is why it is selectively permeable. It is permeable to non-polar uncharged molecules like oxygen, carbon dioxide and steroids. It is non-permeable to ions and larger polar molecules. And it is slightly permeable to small uncharged polar molecules like water, urea, etc. Coming to membrane transport. Basically, we have two types of membrane transport, passive transport and active transport. Passive transport means which do not require any kind of energy expenditure, whereas in active transport, it uses lots and lots of energy. So in passive transport, you can see that substances move down the concentration gradient. It moves from high concentration to low concentration. For example, Brownian motion, or which we can also call it as zigzag motion. Coming to active transport, it uses energy to get substances across a membrane. It moves against the concentration gradient. Endo and exocytosis are the examples of this kind. Coming to diffusion, movement of substances down a concentration gradient is referred as diffusion. The different factors on which it depends, they are steepness of concentration gradient, temperature, mass of diffusing substance, surface area, diffusion distance. I'll tell you one simple example by which you can understand that what actually diffusion is. For example, someday I have used a very well smelled perfume and entered into your class. So as soon as when I'll enter from the door, the first benchers will be feeling the smell of the sweet fragrance of the perfume. It will take few seconds to reach to the last benches. Why so? Because it took some time for diffusing the particles. So the particles moved from higher concentration to lower concentration until equilibrium is maintained. This is what is diffusion. Coming to membrane transport, you can see simple diffusion and facilitated diffusion. Substances move freely across the cell membrane. For example, non-polar, hydrophobic, small uncharged polar oxygen, carbon dioxide, nitrogen gas, fatty acids, steroids, fat-soluble vitamins, water, urea, small alcohols, and many more. Coming to facilitated diffusion, it uses a membrane protein to hitch a rod across membrane. For example, two polar or highly charged molecules move across this. This is diagram you can see. The lipid bilayer plasma membrane, you can see small non-polar molecules, cytoplasm and extracellular fluid. This is how it looks while transport. Coming to membrane transport, types of facilitated diffusion, the topic is channel mediated. Channel mediated moves down the concentration gradient through a channel protein. Rather, channels can be of two types. One can be a leak channel and the other is a gated channel. Leak channel means always the channel will be open for all. But gated channel means it depends upon the type of stimuli or the type of responses the cell is getting. Coming to the other part, see, this is how carrier mediated moves down the concentration gradient. Simple sugars or carrier proteins are involved in this process. For example, glucose, fructose and some vitamins move across. Coming to osmosis, it is a special type of passive diffusion. Water moves from high to low concentration. So low solute to high solute concentration. Water can move in two different ways, between the lipid molecules and through aquaporine channels. Aquaporins are nothing but water-filled channels. Water moves until equilibrium reached on both the sides of the membrane. It uses hydrostatic and osmotic pressure definitely. Coming to the tonicity, we have three types of solutions based on tonicity. First is isotonic, hypotonic and hypotonic. Isotonic means same amount of water on both sides of the membrane. Hypotonic means more water and less solutes. Hypotonic, less water and more solutes. This will be clear by my next slide. See, you can have a clear view. Hypotonic, isotonic and hypotonic. Hypotonic means water is moving out of the cell. That means X osmosis occurs in hypotonic solutions. Isotonic, no net movement of water. You can see the water molecules are entering as well as it is moving out. That is why you can tell that no net movement. Hypotonic means water is entering inside the cell, that is end osmosis. Here you can learn two more different terms, plasmolysis, flaccid, turgid. If in hypotonic solution X osmosis occurs, that means plasmolysis of cell occurs. Isotonic, the cell is maintaining its flaccidity. In hypotonic, the cell is maintaining turgidity by end osmosis. Coming to active transport, it requires lots of expenditure of energy as I told you earlier. Movement of molecules across the membrane against the concentration gradient requires energy. The various sources of energy from where the cell gets by hydrolysis of ATP, that is adenosine triphosphate, energy stored in ionic concentration gradient. For example, ions, amino acids and monosaccharides travel by this. Coming to the last part, primary active transport. Energy from ATP changes the shape of the carrier protein and uses 40% of your ATP for this. Okay, for example, sodium potassium pump. So one hot question I have for my students. Cyanide turns off ATP production. Why is this bad? I want the answer to be find out by my students. Coming to the second hard question for you. What important process uses a proton gradient? You can refer to the slide for the answer. Hint is given here. You just have to tell what important process uses a proton gradient. Coming to the very last part of the slide, bulk transport. Here we'll be describing about the vesicular transport. Two types, endocytosis and exocytosis. Endocytosis means bringing materials into the cell. Three types are there: phagocytosis, solid materials; pinocytosis means non-specific fluid; receptor-mediated means specific materials are present. Exocytosis, releasing the materials out of the cell. You can also tell it as cell vomiting. So this is all for today. I hope you have enjoyed. Please do solve the hard questions for me. Thank you, class. Enjoy your